Welcome to Click for IB Biology. We're in topic D 3.1 reproduction, and we're now going on in this video to look at the process of spermatogenesis, which is the production of viable sperm cells. There's a PDF that you can download that's linked in the description. Add your own notes to that PDF as we go along. Then why not take the 10-question quiz and check your answers, and that's a great way to evaluate your own learning. The structures that we're going to be considering where the main events of spermatogenesis occur are, of course, the testes. And the testes, as you will recall from a previous video, are under the influence of the hormones luteinizing hormone and follicle stimulating hormone, both produced in the anterior pituitary. And whilst they initiated puberty, they are also responsible for the reproductive fertility of sperm production in males. Now, the male testes begins, if we work backwards into the structure, we have the vas deferens. Sperm cells will be coming out of the vas deferens. If you recall the male reproductive structure to the seminiferous tubules and the prostate. The epididymis follow this tube backwards, and we end up in a structure known as the epididymis, as shown here. And this is where mature sperm cells are formed. And then if we follow this backwards, we find that we're actually in a branching system of many tubular branches. Each one of those branches is known as the seminiferous tubule, and you can see that this is in cross-section here, like so. So this is just one of very many seminiferous tubules in cross-section, and in fact, in the center will be the mature sperm cells, and they're going to develop from cells on the outside here. And this is the actual topic of spermatogenesis. Importantly, within the sections of the testes, we have, of course, blood vessels, which are bringing in the luteinizing hormone and the follicle stimulating hormone. And the particular targets of which we're interested in here are tissues like this, which are the Leydig cells, and also the cells of the seminiferous tubules themselves. So let's take a look at the influence of LH and FSH on sperm production. And this requires, to a certain extent, to correlate with our knowledge of meiosis. So the hypothalamus, if you recall, is producing the gonadotrophin releasing hormone, which in turn stimulates the anterior pituitary. And the anterior pituitary will be sending follicle-stimulating hormones to some cells in the seminiferous tubules called Sertoli cells. At the same time, the pituitary will be targeting the Leydig cells with luteinizing hormone, and it's this luteinizing hormone and from the Leydig cells that actually produces the testosterone. Sertoli cells, under the influence of FSH, produce a protein molecule called androgen binding protein. And the function of androgen binding protein is to concentrate testosterone. You'll recall that the pituitary is secreting FSH, and this targets in the outer region of the seminiferous tubule. If I just draw a quick cross-section here. So these are cells in the outer region of the seminiferous tubule. So this is drawn in cross-section, of course. We're in the outer area here. And these are called Spermatogonium, and these cells are actually diploid. And FSH will initially trigger mitosis such that the Spermatogonium cells produce many primary spermatocytes, which are also diploid. So meiosis has yet to occur. The Sertoli cells, which are in close proximity to these Spermatogonium, will produce their antigen binding protein, And this is going to highly concentrate the testosterone in the location of the primary spermatocytes. And this initiates meiosis 1, the first meiotic division, in which the pairs of homologous pairs are separated. And if you recall, we correlate that with the concept of reduction division, the separation, if you like, of the alleles of genes. That first meiotic division with the concentrated testosterone leads us to primary spermatocytes, which are indeed haploid. And so as we go through these divisions, the lumen of the testes here, we're progressing inward. And so our spermatogonium would be in the outer section. Our primary spermatocytes would be slightly deeper in. And of course, in turn, the secondary spermatocytes would be in this location here, all surrounded, interestingly, by these Sertoli cells. And so the Sertoli cells are still concentrating the testosterone, and this will initiate in the primary spermatocyte, which is already haploid. It will then go through meiosis 2, the separation of the chromatids. And this results in the haploid spermatid. Still under the influence of testosterone, concentrated by the androgen binding protein of the Sertoli, will then go through differentiation, in which the spermatid cell will start to develop the characteristic structures of the head structure and of the tail structure of the sperm cell, with its tubulin fibrous proteins in the center here for mobility, and of course the mitochondria are in here. The nucleus, the haploid nucleus, is in the head of the cell, like so. So this is the influence of LH and FSH on the production of ABP and testosterone in spermatogenesis, the production of mature sperm cells, which are known as spermatozoa. And those spermatozoa would be here in the lumen, and they would then move to the epididymis to mature. If we look at this in a more pictorial form, what I wanted to emphasize here was the close proximity of this huge Sertoli cell, which is concentrating the testosterone around the cells which will ultimately become the sperm cell. But if you follow the sequence here, it's exactly the same as in the previous structure. And to correlate that to an actual image, we begin out here is the Spermatogonia, and that's equivalent to out here on the outside of the seminiferous tubule, and the events which then occur progress towards the lumen, eventually with the spermatozoa in this location here, before they are moved to the epididymis prior to their potential release. And so that's the production of sperm cells. We'll go on now to look at the maturation and release of egg cells in oogenesis. That's in the next video linked here. Chance to subscribe to the channel, always greatly appreciated, really is. And then on the right hand side, the playlist, all of our videos in D3.1 reproduction.